Good morning. It's November 17th, and this is your daily brief in science. Here's everything you need to know. Artios Pharma has closed a $115 million Series D, oversubscribed, to speed up its DNA damage response cancer program, including alnodasertib and ART-6043. The round was led by SV Health investors with new investors, RA Capital Management, and participation from Janus Henderson investors, along with existing backers. The company plans a Phase 2 randomized trial of ART-6043, a POL inhibitor in BRCA-mutant, HER2-negative breast cancer patients eligible for PARP inhibitors, following encouraging Phase 1/2a data presented at ESMO in September 2025 data. Greg Smith of IP Group called Artios a leading example of UK life sciences strength with real commercial promise. A cautionary disclaimer about investment advice and cryptocurrency volatility frames the article as marketing context. Since August, Michael Andriole has been CEO, succeeding founder Niall Martin, and he's steering late-stage development and commercialization. Andriole, along with investors from SV Health and RA Capital, stressed advancing late-stage development and building a commercially oriented company. Industry observers from SV Health and RA Capital highlighted Artios' differentiated DDR pipeline and potential for first-in-class therapies in DDR. Artios focuses on exploiting DDR pathways, with emphasis on ATM-deficient tumors where alnodasertib has shown durable responses across 8 solid tumor types. Andriole warned that the Series D accelerates registration pathways for alnodasertib and ART-6043 and broadens DDR development for high-need cancers. Executives and investors noted the milestone would accelerate regulatory pathways and expand DDR development in pancreatic, colorectal, and breast cancers with high unmet need. SV Health investors signaled long-term support for Artios' late-stage development and growth, reflecting confidence in the company's trajectory. A potential clue to long COVID lies in blood clots and immune traps, a combination that could explain persistent symptoms. In a study published in the Journal of Medical Virology, blood plasma from long COVID patients contained microclots that were more numerous and larger than those in healthy controls. Researchers found nets, neutrophil extracellular traps, embedded inside these microclots in long COVID samples, a finding that could make clots more resistant to the body's fibrinolysis process. The study analyzed blood from 50 long COVID patients and 38 healthy volunteers across France and South Africa using imaging flow cytometry and fluorescence microscopy. An AI algorithm distinguished post-COVID samples from controls with 91% accuracy based on microclot and net features, suggesting these signatures could serve as diagnostic biomarkers. The AI analysis used anonymized samples and reinforces the potential diagnostic utility of microclot and net patterns for identifying long COVID. Previous findings also note an increased risk of osteonecrosis of the hip after COVID-19 recovery, indicating broader post-recovery complications. The researchers point to a vascular immune axis as a possible target for treatment, moving beyond antivirals to manage long COVID symptoms. Montpellier Cancer Research Institute and collaborators say the microclot-net interaction could drive systemic changes contributing to ongoing symptoms. Taken together, the combination of microthrombi and net structures could become an objective biomarker for post-COVID syndrome, aiding diagnosis and understanding. A new study on the Jungarian hamster uncovers the functional genetic mechanism behind the timing of seasonal behaviors, providing the first direct genetic evidence in this area as published in eLife. Researchers map the sequence of gene expression changes that control long-term seasonal physiology and behavior, offering foundational insights into how seasonal timing is set in mammals. The work highlights DO3's role in sustaining and terminating the annual cycle, with spring resumption of summer-like physiology aligning with the emergence of mammals in spring. They observe higher DO3 activity in animals near the poles, indicating latitude-linked differences in gene expression related to seasonal physiology, and note that DO3 is evolutionarily conserved across mammals, including humans, showing higher activity in temperate regions and lower activity near the equator. Active DO3 expression is particularly observed in polar adjacent populations, reinforcing the connection between latitude and gene activity. The study, led by Professor Tyler Stevenson at the University of Glasgow, identifies DO3 as the driver of seasonal behaviors such as hibernation and migration in mammals, and states that DO3 must be active to trigger winter dormancy. The full citation is Callum Stewart et al. Hypothalamic deiodinase type 3 establishes the period of circannual interval timing in mammals, 2025. DOY 10.7554/E Life.106383.2. Understanding DO3 expression patterns could illuminate annual health mechanisms and potential links to seasonality-related illnesses. A coronal mass ejection has been observed escaping from a star outside our solar system, a finding that could reshape how we think about space weather around exoplanets. The event was identified by a Type II radio burst seen with low-frequency array, indicating a massive ejected plasma and magnetic field, and was contextualized with XMM-Newton data on the star's temperature, rotation, and X-ray brightness. This is the first confirmed coronal mass ejection from a star beyond the Sun, detected on a red dwarf about 130 light-years away. Low-frequency array applied new data processing techniques to pick up the radio signal, while XMM-Newton provided measurements that help interpret the eruption's nature and motion. The discovery resolves a 30-year debate about whether stellar coronal mass ejections can escape a star's gravity and magnetic field and travel through interstellar space. It also demonstrates a direct observation method for stellar coronal mass ejections, using a combination of radio and X-ray data. The study suggests that smaller stars may experience even more extreme space weather than the Sun, with potential implications for the atmospheres and climates of orbiting exoplanets. A new analysis method called radio interferometric multiplexed spectroscopy, or RIMS, was used to interpret the radio signals and confirm the coronal mass ejection's origin. The finding highlights the importance of extrasolar space weather in shaping planetary atmospheres and the habitability conditions of exoplanets. Researchers say repeated coronal mass ejections could influence atmospheric loss and climate on planets around other stars, underscoring a new frontier in stellar space weather research. BioRestorative has FDA Fast Track designation for BRTX-100 and plans an accelerated BLA pathway with a Type B meeting with the FDA planned for mid-December to discuss timelines. The meeting will explore moving from Phase 2 toward a Phase 3 trial to support a BLA, using the Fast Track status that's already in place. Chronic lumbar disc disease is a common costly condition with limited disease-modifying options, and current therapies mainly relieve symptoms rather than reversing degeneration or restoring cell homeostasis. There are no approved therapies shown to reverse disc degeneration or restore intervertebral disc cell homeostasis in CLDD, highlighting an unmet medical need. Background details include the CLDD context, BRTX-100's production process, and the trial identifier NCT04042844 registered on clinicaltrials.gov. Phase 2 analyses to date report consistently positive safety and efficacy signals for BRTX-100, according to BioRestorative Management. BRTX-100 is an autologous cell therapy using a patient's cultured mesenchymal stem cells from bone marrow, designed for non-surgical treatment of disc disorders, evaluated in a Phase 2 randomized, double-blind, placebo-controlled trial across up to 16 U.S. sites, with up to 99 subjects. The therapy targets disc degeneration-related back pain and is intended for outpatient injection into the damaged disc, with Phase 2 trial parameters including randomization and blinding. The press release states plans to update investors after receiving FDA meeting minutes and includes standard forward-looking statements and investor relations contact information. BioRestorative's broader portfolio includes ThermoStem for obesity and metabolic disorders and a biocosmeceuticals platform, with BRTX-100 as the lead candidate in the Disc and Spine program. No numeric Phase 2 data or endpoints were disclosed in the release. The accelerated pathway depends on FDA feedback and the written minutes. BioRestorative emphasizes consistently positive safety and efficacy signals from Phase 2 for BRTX-100 and intends to push for an accelerated timeline into Phase 3 to support the BLA. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.